by request from one of the panelists, and that is to really just get some comments about the title of our discussion, the future of the personal genome. We've heard, and you all now understand it before, the inexorable decline in the price of genome sequencing, accuracy will improve, tools to interpret the genome will improve. I think it was Jamie who said we'll all be sequencing genomes in our iPhones in our pockets or something. So, you know, what's life going to be like when personal genomes are that abundant, that easy, that cheap? How's that going to impact life, medicine, healthcare? Because that day's coming, right?
So I think that it doesn't take much extrapolation to see that when there are 7 billion of us each going to get our 6 billion base pairs. But that's just the start. That's 10 to the 19th base pair start, because a lot of our body changes from day to day. So we're measuring, and so in the Personal Genome Project, we're already modeling this with actual measurements. You can measure from the blood a variety. You can measure transcriptome from a variety of available tissues. You can measure your microbiome from a variety of different body sites, your immunome, your response to it, and exactly which antigens it's seeing in the world and autoantigens it's seeing in your body. And we've already published some of this. And you're with your cell phone on your body. You will be monitoring the environment as you walk into it, and you'll be tweeting to your entire set of friends that you just walked into a big cloud in such and such a cafe of H1N1. And so suddenly the cafe will empty, and who knows what happens next. So that I think is the future of personal genomes.
Jamie's speechless for us.
I'm running out of the cafe. So a slightly flip, but not entirely flip, response is, as far as I can tell, every major technology is first adopted by the porn industry. So I would begin to look at the implications. Video streaming, e-commerce, yeah, I could go on. I mean, you know.
Please don't.
I did go to MIT anyway, but but no, the less flip. I mean, I think George is right. I think the sort of the comment about you know, what did you say, read once and read once and
sequence once,
read forever. I actually don't think this is a fight I have with George and others regularly. I don't actually think, other than cancer, DNA is actually that meaningful. I think about 1% of us are going to have really important information from our DNA. 10% of us are going to get actionable information, and the rest of us are going to get not so interesting information. And then we can argue about the numbers, and George will tell me that's still 30 million people, and that's an accurate statement. But U.S. citizens.
And it might be you.
It might be me. I'll listen. Maybe if you get your butt again. But but the part about, but I do think that the concept of state is really important. I think that looking at transcriptome and the immunome, now you want to cup, you want to buy it. I think those are really critical issues, and what I'm afraid of is that the quality control in science and how well we do these things, and exactly the issues that Michael raised about how you do this properly, are really going to set us back. I think that if you look at you know, gene shifts, the first 7 years were basically a crap wash of bad quality control, and we're not learning in science and biology and medicine how to do quality control early on. And I think you know, we have to get that in line really quickly, because these things are going to move too fast and be too important and be too critical. But the future to me is, you know, I'm imagining Surrey, and you know, she'll wake up in the morning, she'll say, you know,
You're talking iPhone, just
iPhone here. That's the Surrey's the nice Apple voice thing. Is she's going to say, you know, according to where you are, listen, if you do 20 push-ups before you go to work, you're going to drive this number up, and it's going to control your diabetes models to the point where it's great. She won't tell me any of this. She'll just say, do this and you'll feel good. And by the way, you didn't take your aspirin, and your blood's a little thick, so let's fix that. And maybe you need a little bit of medicinal. Let's invent some new substance. Provigil to focus to finish the book you're working on, because it's going to bring your amygdala. Where are our friends at Indigo here? They have a good drug, nicotine agonist. You need that to get some focus into your life. I think those kinds of technologies, I think it's going to be direct advice, and it's not going to be about illness. It's going to be about wellness. It's going to be about, to be really personal. I mean, look, my mom and dad, who I love completely dearly, and came to hear me say a few embarrassing things at MIT tonight. They're both in their 70s now, and I have young children, and I want them to, I want my father to keep working to pay for my children's college. So and my children are 1, so that's going to be a little bit of time, and or some of them are, and so I don't have a molecular measure that can tell my dad the level of adipose fat in his muscles. We go see a personal trainer together every Wednesday, and how that will affect all the things that drive his health. And I want that number, because I know what he's a numbers guy. If you just give him a number, he would fix it. So I want the number. I want the number that says, do this, and Surrey can give it to us. And he knows what it is.
Colin, the question?
Yes. So I guess I look at things from a very different vantage point, in that $1,000 genome I take to mean it's really cheap to measure all of the molecular stuff going on in the cell. And I see it as a chance to finally predict the behavior of complex systems that we've wanted to do for a really long time. Right? The computational methods, machine learning, and modeling and simulation techniques have been around for a long time. We know that there is some underlying genetic network of disease. Mike showed a little protein pathway involved in some of the more popular cancers. That's only a small slice of the true complexity we're dealing with. If we really want to be able to predict the behavior of systems and get at some truly predictive medicine, I look at the availability of the $1,000 genome as a chance to finally get the data that we need to turn that data into computer models of disease. And I think the implications even outside of healthcare are massive. I mean, if you think about energy, and if you think about food production. And so that's really the way I see it. There's a lot of other fields of study where there's been sufficient data that they have truly predictive models. And I'm talking about like 80–90% predictive in terms of being able to intervene in someone's care.
So if we think about it, I'm going to jump out 50 years, because 5 years, 10 years, 20 years we can debate. 50 years, it's something is going to be there that's very different than today in 50 years. Whether it's $1 or $5 or $20 for your genome, it's certainly going to be in that range. On the oncology side, listen, we have a glimpse into the future. There's no doubt about it. In 50 years, the vast majority of cancers are going to be treated with very specific, individualized therapies. We're going to do the complete assessment of each tumor. We'll be able to link it to a targeted therapeutic, but more appropriately, each person is going to be treated with a cocktail of therapeutics. Again, HIV gives us a glimpse into the future. One protease inhibitor didn't do it, so better make it 3. And that's where we're going to be with cancer. It's all about, the pharmaceutical companies understand it today. They're thinking through the approach of how they're going to develop these cocktail-based, take a cocktail-based approach in the future, because it's not easy. It's not easy to design a trial and do it today. It's not easy to think about getting through the FDA. But clearly, that is going to be the direction in 50 years. In 50 years from now, it's not going to be 100% of cancers, but clearly, whether it's 70% or 80%, the lion's share of cancers will be treated at the individual level, and they will be treated with cocktails of drugs. And I'd be hard pressed to think that it's going to evolve any other way. When it comes to the personal genome and just having this information on your credit card, I think about it a little bit differently. I think about a lot of things a little bit differently than Jamie, which is okay. Think about the iPhone. Steve Jobs. As I'm listening to you guys talk, I was thinking about Steve Jobs 25 years ago, and he had envisioned where the world would go with all this technology. 25 years ago, on my best day, I could not envision having an iPad or an iPhone in my pocket right here and having access to all the information that we have access to. But it happened, and Google came around, and Twitter came around, and all this new technology comes around, and it just kind of happens. It's just like, if you think back 10 years ago and you think these companies didn't exist, and now they dominate the landscape today, when you think back, retrospect, well, that's pretty darn impressive. You have $10 billion and perhaps $100 billion company that didn't exist 10 years ago, yet they're having an enormous impact on our lives today. But I don't know, I still get up out of bed in the morning, and instead of listening to an old clock radio, one of my Apple devices goes off, and I'm checking email throughout the course of the day. But life doesn't feel all that different. And I suspect with a personal genome in my wallet, life is not going to feel all that different. My day-to-day transactions in medicine will be different. I guess I'll be fortunate enough to be alive in 50 years, so I guess I need to think about that. But things just happen. And I don't think, we're not going to hit a tipping point one day. We're going to hit a tipping point that's, over the course of 10 years, and we look back and say, my gosh, can you believe that everything is right here, and these are the things that I can do with my credit card when I walk into my physician's office today? Or probably more realistically, these are the things that I can do on whatever the device is of the day without going to the physician's office to make these changes.
Okay, it's your turn. Lots of hands. So we'll start over here. Speak nice and loud. Bell mics.
Right. I want you guys to test my hypothesis that this technology was to destroy the pharmaceutical industry. And I'm going to run it by you this way. I want Michael to tell me what you say to a biotech or a pharmaceutical executive when you present to him that he would have to create up to 31 different drugs on a one-to-one basis to solve a specific cancer. Colin, I want you to explain to me the technical challenges around doing computation around an up or down, just up or down, of each of those genes. It would be essentially a 2 to 31 computational challenge. James, I want you to tell me the consequence of your form of patients that constantly complain about drugs and their adverse events. What do it mean to them to have a cocktail with as many as 15 to 20 different compounds, each with their own adverse event profile? And what the consequence on your profile, patients like me, that would be. And George, I want you to explain to me how you would explain to the doctor what I just asked the other three guys.
That's like my slides, 7 slides in one.
If we're going to get comments from all 4 of you, we need to be quick to give up.
Yeah, I'll give a very simple answer. I tell the CEO of that pharmaceutical company something he already knows, and that is you better be really nice to the FDA to figure out a way for them to get their acting gear to work through these things. Start there.
My answer, the dimensionality of the space of possible connections is massive, but it is not the limiting factor. The computational power is here. A lot of that was invented even here at MIT. That's not the limiting factor, it's the data.
I don't, actually, I think the opposite is going to happen. I think the pharmaceutical industry's role in society is to deliver effective drugs for known targets. The reason they're in trouble right now is the number of targets are going down, and what this is going to do is reinvent discovery in a way that gives them much more effective targets to work for, and the trials will be much smaller, much more cost-effective, much more focused. I think they're going to make out like gangbusters.
I actually agree with that.
Yeah, so do I.
By the way, they are my client, too.
And I think pharmaceutical industry is going to do fine, not necessarily with small molecule drugs, though, and not necessarily cocktails. And so what's going to be different is there's now phase 2 clinical trial from Sangamo where you can delete both copies of the CCR5 allele, which is the HIV receptor, and you become resistant to HIV and a variety of other pathogens, cure for AIDS. That's Sangamo. And then David Baltimore and others are investing in their time in getting rare antibodies from the population that are neutralizing HIV and then giving them as antibodies, not as vaccines to individuals. Those are two examples where we're not talking about small molecules anymore. It's a brave new world that will be highly personalized.
Right. I just want to make one comment. The immune system does this every day.
Exactly.
Well, actually, but riffing on this, I think it's really interesting, and I don't know how many people are in the finance industry. How many people are modeling the risk that we cure diabetes, type 1 diabetes, on the financial impact of the companies in the space? Because I think it's going to go down in the next 10 years. Totally. I mean, like, end of story, fixed. And, you know, how many people are thinking about what happens to that $20 billion industry when that happens? I don't think finance models that kind of instruction.
Steve.
Thank you, Kevin. I'm Steve Dickman from CBT Advisors, and I just wanted to bring up a quotation I heard from Lee Hood at the Personalized Medicine Conference in Boston in November. He said, if we in the United States do not give our personal genomic data to society, China will beat us to it.
Yes.
There will be Chinese sequencing of the Chinese population in a way that will become a societal threat to the United States. Now, if you read my tweets at CBT Advisors, I tweeted, so? And I want to ask the panel, so what? Is that a real threat, and what does that actually mean?
I would love to have international cooperation on getting our genomes. Yeah, that'd be great.
Actually, I have some answer. I just went to St. John, BGI.
Yes.
Yes. They have 6,000 people, and the company feed them breakfast, lunch, and dinner. And they sequencing day and night. You're right. The data will be out. It's a no charge.
Anyone else? Thank you. Anyone else want to comment on?
Well, Colin raised privacy, so what are
Well,
the
that's it, Joe. It's
implications for
government
privacy?
mandated versus what we do here in the U.S. And so, you know, it's a lengthy conversation, but it's one thing when you can government mandate something.
The U.S. actually government mandates 40 tests for every newborn in
Sure.
hospitals, so
Sure.
we're not that far away.
Gentleman in the scarf. You?
Yeah, thanks a lot, guys, for a great discussion. If the old tipping point was the $1,000, what the $1,000 unlocks is the number of patients you can sequence. So I guess the question for all of you, one way or another, is what is the N you need? You know, Michael, it's for you if it's for tumor normal. Colin, for your statistical models. Jamie, to match your genomic stuff. George, to take the PGP to however big it would be. What is that number that takes us to an analysis level that's really meaningful?
7 billion. But keep in mind, we're all an N of 1. Yeah. And that's very important. Causality beats correlation any day.
I actually think that the question's wrong. I again, don't particularly think that genomics is going to be the tipping point. I mean, the GWAS studies, and, you know, George...
Long mile.
I know. So, George, we can skip into technical arguments about how much more we're going to get out of the full genome. I think the tipping point is actually a different number that I'm looking at, which is if I draw blood from a patient and I want to measure with real coverage all the RNA, fax all the cell surface markers, look at a significant number of proteins, look at all the metabolites, and begin to do some of the very, you know, nascent technologies that can look at the immune system. When that number is below about $2,000 a sample, which I think it's getting close to this year, then you give me 10,000 people and we do that over a few years, and we will know more about the molecular health of human beings than has been learned in the history of the NIH. And they have no hope of funding that, so.
And what we need, it's not necessarily the sequencing horsepower. It is, in fact, you know, patients like me. It's the clinical data. It's the follow-up. It's understanding what the impact is as we utilize this information to either change our lives or to treat cancer differently. So what? Well, there's only one way you can figure out that so what, and that is to track patients over time.
Lady in the red.
Yes, Michael, you said earlier that gave an intriguing number. The $1,000 equates to 18%, only referred to 18%. Can you talk a little bit about what the other, you know, costs are?
It's the stuff before and after sequencing. So, and really that is, it's everything that we do to prep the specimen, to get it ready for the sequencer. So it's all the steps in the laboratory, in the wet lab, from extracting, prepping the tissue, extracting the nucleic acid, and bringing it through the processes.
So that's the $750 that people talk about.
Well, I won't break out the exact. It's a component of the total cost, and then on the back end, it is in fact the analysis of the data. And it's taking the data, once you have the genomic data, it's marrying that up to the actionable data and to the medical data, which over time, a good bit of that can be automated, but today, a lot of it is still manual. So, there's a bit of sarcasm in the answer. It's everything before and after, but that's the honest truth. It's challenging to take these specimens, prep them, extract nucleic acid in a robust way so you have enough of it to bring it through the process onto the sequencer, and then on the back end, from the sequencer, we don't come up with 3 data points. And we literally have millions of data points that we then have to sort through and turn it into the clinically relevant, our definition of clinically relevant. And then we have to take those clinically relevant data points and annotate them with real information that's useful to the physician. So it's the whole process.
I just want to go on record as saying I'm a little skeptical of that model because, there are 2 companies delivering $4,000 genomes that involve more or less the same DNA prep. They are only doing it 40x coverage, but if you're doing it 1,000x coverage, then the DNA sequencing becomes a bigger fraction of the variable cost, not a smaller fraction. So they do interpretation and they do DNA prep, and the only thing that doing 1,000x would do is blow up the sequencing cost more. So I'm listening.
And the last thing I'm going to do is get into technical discussion with George. And so I may not be smart, but I'm smart enough to know not to do that.
We can take questions. Questions about
actually put a question to that. So in this world where the cost of sequencing is dropping to the bottom, right? Where literally I think the last number we heard is $1 per genome perhaps someday. What sort of business models do you see would be viable, and what sort of companies would we see actually in the future to sort of take advantage of the personalized medicine?
George, what companies will you be launching when we have the $1 genome?
Well, everybody, 7 billion people have their genome, and you'll be interested in what changes from day to day. So it's the immune response to the microorganisms, the allergens, the foods that you eat. So you're going to be constantly monitoring your environment and your body's response to it. And even if that's $1, you're still doing it a few times a day, it can be real money.
We haven't talked about the microbiome this evening. Is that something
It used to be that you would assess your environment with a questionnaire. What did you eat for the last 3 months? Did you get, sniffles? Now it's going to be, you got rhinovirus such and such on this day, and you had this immune response to it, okay? So the Personal Genome Project, we monitor 5 body sites as often as we can afford.
Yes, sir.
You didn't say anything yet about the epigenome and expression levels. Do you want to speculate on how this enters into the picture that you just described?
Well, several of us have mentioned that, but by a different name, which is RNA or transcriptome, or all these things are relevant. The real problem with epigenome is your genome you can get from almost any part of your body, your saliva, your blood, whatever. The epigenome, you have to get it from the right tissue. Now, if you have the beauty of the cancer is you get a piece of the cancer and you know that that's what you're supposed to be analyzing for genome or epigenome. But for almost everything else, some of those tissues are very hard to get at. So if you have psychological problems, for example, you don't really give up the appropriate tissue for that.
2 questions in the back there. Either one of you.
My question to you is, we're talking today focused on prognostic nature or to look into patients who are actually suffering from cancer. But there are folks who survive cancer and are healthy. I'm curious to know if any data could be mined or generated from people who suffer cancer, were cured of it, that could be applied to the previous folks I just mentioned.
I want to hit this in a couple ways. I mean, if the tumor's gone, then you've lost some important information about, what they had. And I think this really gets to an issue that medicine has today, which is essentially what is the true standard of disease. So I have some friends that are doing biomarker discovery in a variety of areas. And what's interesting is when they work in a cancer that has a very high true standard, like pancreatic cancer, where the diagnosis is probably 97, 95% accurate, they can find pretty strong signatures for these, looking at blood and with a variety of technologies. When they look at a cancer where the true standard is actually not very good, like breast cancer, what you find is that the samples, the diseases, are confounded with people that don't actually have cancer. And as George said, one of his, which cell was it you cloned?
Induced pluripotent stem cell?
Yeah, but it was a skin cell, right?
Skin.
So George's skin cell had a single cancer gene that was mutated against his background genetics. Was that real? Was it a consequence of the cloning? We don't know. We don't know the answer to those questions.
It was a consequence of the cloning.
What's your margin? What's your margin?
We have the original fibroblasts and they were unmutated.
Okay, I'll give you that one. I made the mistake. It was a trap. I went totally. But I think the real issue is that we really don't know what disease is, and that's breast cancer. I mean, I think if you go to things that are much more vague, like fibromyalgia, Crohn's disease, or multiple sclerosis, we're looking at a situation where, I think this is the hardest thing for medicine, because to your question, we're talking about the redefinition of human disease, period. Cancer is no longer tissue-based, it's genetics. Foundation, it's gone. I wouldn't even build a tissue-based approach to cancer any longer, and no one is. But the definition of neurological disease will be at the molecular level, the definition of all these diseases. And so I think it really is about collecting good data going forward, not. I think we need to understand what these conditions are, because I think we fool ourselves into thinking we understand disease.
That's yes, gentleman in front of the previous question.
So to follow up on a couple comments you both made about data analytics, and particularly you, James. I looked up what your company does real quick, so I have a surface knowledge of what you do. But presumably you've taken some profile, that gives you some structured data. You have a whole bunch of feedback that's coming in, and you're running some NLP sentiment analysis on it, and you mentioned too, so you already have multiple sources, and you're extracting some information out of this. And then, Colin, you talked a little bit about a never-ending trial, so to speak. So this is not to belittle the challenge out there, but something very similar has already been solved. So there are other highly complex systems out there outside of biological systems. So financials have solved this already, right? You take multiple input points, and it's not just, when you talk about ongoing trial, Bloomberg, for instance, does this. They do this with summary data, and depending upon how deep and how high you want to climb up the stack, from raw data to information to intelligence, analytics, and so on, you can go back and you can go deep. So I'm having a hard time trying to really grasp, when you start talking about freeing of data, there are a whole bunch of efforts that say, if it's NIH funded, it needs to be on data.gov. What is really preventing from data to get liberated that's coming out of these sites? Is it just a format, a standard, or is it something else?
I think a lot of things. You think you have incentives that aren't necessarily aligned. You have a lot of academics who are trying to get to glory and publish papers. That's the world they live in. They do not share their data. And there are issues around IRBs, approving access to clinical samples. And certainly when you get to the healthcare system, so health insurance companies, pharmacy benefits managers, there's HIPAA laws that make this really difficult. I was down in DC, Todd Park, who's the CTO of HHS, organized a little gathering around Center for Medicare and Medicaid data users. And that was one of the big major themes, was the fact that right now it takes 6 months and about $500K to get a slice of data from the government-funded healthcare system. Right? That's a crazy amount of money and time for the number of people who want to use it to mine the data, to actually figure out what interventions are wasted when versus what works, and so on and so forth. So we're not close to having the kind of data liquidity that is taken for granted in the financial trading world and in the online advertising world. 3.5 years ago, our company actually spun off a quant trading effort called Pellini Technologies. And seeing that thing grow up, it was really interesting to see relative to how long it takes to get data out of a clinical trial or to get data from real-world studies, how quickly these guys could make models, generate predictions, and test them, right? If you have a predictive signal about a certain US stock or a certain currency, you can trade it next week, you're going to get feedback right away. In the healthcare system, it takes so long, and there's never enough data. Even in the early days of genomics, the best pharma companies, in terms of genomic data, when you actually got under the covers, they really didn't have that much. Merck, I'll name names, Merck didn't have that much data, and they were probably the best in the industry going back to the early 2000s. And so the point is that there's a lot that needs to change in order to enable the data that's generated from everyday medical care, whether it's deeply molecular or not. That's the biggest challenge to actually making all of this data actionable and actually making a difference in medical care.
Just a quick point of clarification. Is it not true that personally controlled health records are not affected by HIPAA, just electronic medical records? So
yes.
So in the Personal Genome Project, the way we've cut through that Gordian knot is we've turned all the electronic medical records into personally controlled health records, and those are now on our website for all of you to look at.
That's a fantastic
so it's not that hard.
George. And that does bring up, there is something called a blue button. It's now available in the US. You can now get access to your own health records. There were some legal things that had to be figured out over the past few years. There's now 400,000 people who have pressed this blue button, and their data is there. So right on.
Where did you find the blue button?
It's the VA. It's growing. But actually, there's one other thing that we need to touch on here, because I think I love the finance metaphor. And do you know why we can compare IBM to GE to Ford Motor Company to Pfizer? Because there are these things called accounting standards that everyone has to follow, and there are these things called auditing companies that have to make everyone follow those standards. Now, medicine, the way we would do this is that everyone would get to invent whatever they thought goodwill meant. Everyone would get to invent whatever they thought profits meant, and no one would report what currency they did the transactions in, and then you would expect to be analyzed this effectively in the end. So we need to, in medicine, have those things solved. And what I love, my proof point, this is a great proof point, is if you look at biotechs, when a biotech has a phase 2 study that the FDA has audited, FDA is an auditing agency, that's all they really do. Their decision process sucks, they're very good auditors. The value of that company goes up literally 10, 20, 30-fold immediately as soon as the FDA said that data is real. And the driver for that, to the evidence I could see, is that before that, no one believed anything anyone said because no one audited anyone anything said. And that's the only reason it works. And so if we could bring the auditing back earlier, we can actually develop a functional market. And that's what's missing. There are no standards, there are no definitions. The personal one that really got me was we were doing a community for blood cancers. And then we did this survey. We went out and talked to all these patients and all these clinicians, and we asked them about their record keeping. And the clinicians did not know what they did to the patients because they don't keep version control on what CHOP means. CHOP is a kind of a chemotherapy treatment. So they don't know what they gave people in their own hospitals, let alone being able to tell the patients what it is. So we have a lot of work to do before we can do Bloomberg in medicine.
Let's go into the rapid fire round, about 5-ish minutes. You had one.
I didn't.
You didn't, excuse me. You're right, sorry.
Is there anything important in your world that is compute limited? Not data limited, compute limited?
Yes. You can't exhaustively reverse engineer genetic networks from expression and sequence data exhaustively. You can do it approximately, but there's no way the number of possibilities are astronomically large, not just like billions and billions, but like 10 to the 346. So that's limited.
I would say that he's right, but We haven't done the obvious problems that fall out immediately, and I don't think that the initial problems are computer issues.
So in the context of reverse engineering, George, could you speak to the future of where the genome reengineering may be going?
Right, so the question is about genome reengineering, and I think to some extent the computing problem can be solved by really good data, and lots of it, ironically. It's the more data, the easier the problems become. In terms of engineering, you can test hypotheses with it, and I mentioned already a phase 2 clinical trial that is literally human genome engineering. I don't see, I think that we're also getting gene therapy in retinal degeneration diseases, cancers, and so on. I think we're going to see the 10-year moratorium essentially on gene therapy disappear overnight. There's a zinc finger nucleases, tau nucleases, recombinases. There's going to be a whole lot of them that are highly specific.
Yes.
I'm very intrigued by the question about connecting all this data to meaningful outcomes for patients. As a physician, one of my great frustrations has been the thousands of patients I've treated that hasn't added to the knowledge. And Colin, you mentioned the idea of a continuous ongoing trial. But in order to have that, you have to have a way to collect systematic, accurate, meaningful data that can't be under the rigors of a funded clinical trial, but has to be part of the routine, everyday part of care. And I would love to see a vision for that. I don't see that yet, and that's one of my biggest worries for all this, because there's all this beautiful data, but it seems to fall short in a lot of clinical conditions on an outcome that is meaningful.
I think access to that data has to open up in a major way for it to be useful, which means there's legal issues to getting that data and getting the feedback back to the doctors, back to the providers to actually do something.
I would argue that data doesn't exist. I would argue that you look at health plan claims data, and you look at Medicare data, even physician office EMRs that you can interrogate very efficiently, that data doesn't exist. And so what I'm saying is, how do we put in ways of looking at so we actually can get functional status, pain, disease-specific responses, that kind of thing, because everything so far has to be limited to very high labor-intensive clinical trials to get that kind of thing.
Right. I think for the most part in the U.S. it doesn't exist, but there are places, integrated delivery systems like Kaiser, Geisinger, Intermountain, that have at least the beginnings of that. Like it's once again, it's not fantastic data. It's just claims level, sometimes integrated with the EMR, but it's getting at the beginnings of it. I still think we're years away from having the kind of data we want to need. So I'm agreeing with you. I mean, I'm the last person who's going to say that the data is fantastic and there's nothing holding us back. But I do think there's nothing. The technology is not the problem. I think hopefully that's clear to people, because we do have Amazon.com and other industries where they've been able to get close to real-time data and to learn from it, and to even something as trivial as recommendation engines that does work. Now, the medical system has a lot of issues with it, and dealing with my cousin, the difficulties in her cousin, getting authorization to get her data and so on and so forth, what does that bear? So I think for routine care, it's still many years off, but I think it's the challenges are just human.
Three-word answer. We decided that if we make it easy enough for patients to do it, the doctors might be able to use it. So a couple more years' work, we'll get it for you.
Who is confident they have what it takes for the final question?
Thank you, John. So we've had on one extreme that Mike commented that 20% of 50-year-old technology is done with a lot of errors. The genome is not sufficient for anything probably more than cancer. So to get this integrated omics conclusion and get that out to the mainstream, to the late majority generalist in Kansas, I think really is human generations, 40, 50 years away. Not to take it down to the whole audience, but Steve Jobs didn't have regulators and figures in his brain.
Yeah, so I think to get everything to the mainstream, I would agree with you. To get aspects of this to the mainstream, I completely disagree with you. So not to end on a company pitch, but certainly one of the things.
Damn, you're good.
So I think that's the secret. The secret is finding a way to get this out of the Dana-Farber's, to get this out of the Mass Generals, and I say it with great respect because, but listen, the vast majority of patients in the United States are not treated at Memorial Sloan Kettering or MD Anderson, and 98% of them are treated in their communities. And the trick is to translate this data, not all of it en masse, but pick your project, translate it one step at a time, and we can get it into the communities. I saw that firsthand at my last company. Much simpler technology, but that's kind of that's what we did. Now we have to up it a little bit with a new type of technology. You can't do it en masse, but you can take bites out of it and do it one step at a time.
Anyone else want to comment?
Gibson said it well. The future's here. It's just not evenly distributed.
All right, you've been a great audience. Please join me in thanking our